Hello, I'm Asar Rashid. I'd like to talk to you about a paper, a transcriptomic appreciation of childhood meningococcal and polymicrobial sepsis from a pro-inflammatory and trajectorial perspective, a role for Vegf-A and Vegf-B modulation. In this paper, we're interested in pro-inflammatory cytokines such as TNF-alpha, which leads to the excretion of Vegf-A, which is angiogenic. On the other hand, we're also interested in Vegf-B, a survival molecule, which has various safeguarding effects, such as being cardioprotective. We aim to study time-related sepsis dynamics from a gene expression perspective, and particularly we were focusing on certain pro-inflammatory genes, TNF-alpha, NF-kappa beta-1, Vegf-A, as well as Vegf-B. We looked through the current literature, and using the search strategy as detailed in the paper, we selected 2 temporal and 2 cross-sectional meningococcal septic shock datasets, and also 1 polymicrobial temporal dataset. Here shown are transcriptomic profiles for Quan dataset in A and B above, and EMONTS in C and D below. In Quan, 2A PCA depicts a temporal direction, which by heat map shows distinctive clusters, which we label as early, intermediate, and late states. For the EMONTS dataset in C and D, you can see this PCA spatial representation according to cell type in C, which is on the left, and then on the right, D, it's the comparison of control versus sepsis. The EMONTS dataset is then shown according to heat map, showing distinctive clustering according to cell type in E, and then after ANOVA filtering according to time, the trajectory is depicted in F and its associated heat map showing early, intermediate, and late states. The TTCA gene enrichment is presented here for the Quan and EMONTS datasets according to the KEGG and HUGO databases. In bold are the significantly enriched pathways, and if you have a look at the EMONTS, the KEGG Vegf signaling pathway is highly significant. This is the Brosletto postmortem dataset where meningococcal sepsis samples MSS are compared to acute non-infectious cases, and you can see here that Vegf-B is acutely downregulated compared to NF-kappa beta-1 and Vegf-A, which are upregulated in the MSS dataset. B specifically focuses on the MSS samples only, and you can see in the renal tissue, Vegf-A is highly upregulated in the kidneys compared to the heart, lungs, and spleen. Looking at the EMONTS dataset, you can see here NF-kappa beta-1 shows differential expression in the different cell types, with whole blood being most expressed and less expressed in lymphocytes and monocytes. Vegf-A, on the other hand, you can see it's highly expressed compared to lymphocytes in the blood and monocytes, where in Vegf-B, the opposite pattern is seen, with lymphocytes showing high gene expression of Vegf-B in lymphocytes compared to whole blood and monocytes. Again, in the EMONTS dataset in B, you can see upregulation of TNF, NF-kappa beta-1, Vegf-A, but no difference in Vegf-B in controls versus sepsis. In the temporal changes with the EMONTS, you can see an escalation in gene expression from 8 to 72 hours. Similarly, in D, which is Quan dataset, you can see a differential upregulation in NF-kappa beta-1 at 48 hours compared to 0 hours. This is the Wong polymicrobial dataset. If you look at NF-kappa beta-1, you can see that the 3 subsets, SIRS day 3, sepsis day 3, and septic shock day 3 are upregulated against controls. Moving on to Vegf-A, only sepsis day 3 and septic shock day 3 are upregulated against controls day 3, whereas in the Vegf-B group, all 3 subgroups are downregulated against controls. In conclusion, unsupervised heat plots reveal distinct early, intermediate, and late phases of meningococcal septic shock based on gene expression. Temporal changes in NF-kappa beta-1 gene expression were identified in MSS, suggesting its potential as a biomarker. Consistent upregulation of Vegf-A and NF-kappa beta-1 in sepsis from the gene expression datasets was observed, with Vegf-B downregulation also noted.